All right, hello. Um, well, this is my DNA project. So here is the diarribosome sugar that shows the the black thing that's holding the pieces together. And this is my I forgot how to say that word, but it shows right here that helps to keep everything together. And the nitrogen's bases are like the little bees for like your DNA or something. And the hydrant bonds are the things that be able to hold these beads together right here. So I wrote down like I went I looked up what does these things do to help us. And the hydrogen bonds are important to the human body because it's a highly I don't know how to say that word, atom that produces nitrogen, oxygen, and fluorine. I still don't know how to say that word. Molecules helps to keep the DNA sequence together. Nitrogen's basis helps by building blocks for the helix, diarrhylates, I forgot to say that word, sugar. It defines if some sugar or oxygen was lost. Now on to the cell cycle. The S represents for for interphase because it shows the DNA replicates, and the two G's um, the two G's represents for the growth. G1 represents for growth. G2 represents for growth, preparation, and for cell division. And mitosis is each of these. Okay, each of these points are checkpoints between the cell cycles. Yeah. So the S phase helps the human body by preventing genetic abnormalities, which could lead to the cell dying or getting a disease. This is the picture of the DNA polymerase, whatever that is. Yeah, the double the double helix works by cutting into the DNA into a single strand, so each strand could be copied. The I don't know how to say that word. Creates two molecules of the original DNA, makes a copy. I kind of messed up on the slides, but this is a picture of the double helix, and it shows it split into one. So like all CGs and all that are split into one strand. The double helix opens up, and once the strand is separated, the polymerase copies each strand by using the basic pairing rule. This is a picture of transcription. See, here's the double helix open up. And this is this word right here, I don't know how to say it. It's readings the copying stand so it can each have its own copies. Interrupting. Alright, so it shows that it reads the coding stand, and then once the helix opens up, it goes back to it goes. They combine once again, and this shows the RNA leaving the nucleus.
And that is so true.
How you just go? Yeah, that's it for this. The transcription occurs during a subpith. So, I forgot to say that word. Period of the interphase. The process of transcription is in the stand of DNA is copied into a new molecule. So DNA safety and stably stares genetically material in the nucleus.
True.
All right, this is the point mutation.
Point mutation.
Good. Okay.
This is the point mutation.
Okay. You interrupted my recording. This is a picture of the point mutation that can occurs in a human DNA. So over here is the basic pairs of the normal DNA sequence. Over here is a mutation because on the basic pair it's supposed to be different colors like blue, black, red, and green, and it goes all the way down. But on here, instead, it has the same sequence as because there isn't no three pairs of junior, or whatever that word is called. Gene mutation is the base of species diversion among beetles or any organism. Mutations are changes in a genetic sequence. They occur by different levels and widely different consequences. This is a picture of the DNA polymers 3. It shows each strand is splitting into one strand again, but instead it just keeps going. And over here, this is the RNA. Number 3 is for RNA, and DNA is, I guess, is the number 5. Oh yeah. The DNA is splitting into two, so the polymers can copy each strand, so our DNA can keep growing in case our old DNA molecules dies, and new ones will be ready to be used. This picture is kind of scribbled up, but this is the picture of the translation. And here you have the ribosomes, the RNA, the empty tRNA. And what this says, the translation process incorporates two different animal acids in a proceed sequence dictated by the 3 base codes, built from an alphabet of 4 bases. The process in the ribosome builds the chains that will become proteins. The translation occurs when it's the last step to the DNA to make more proteins. And there you go. That's my definition of DNA sequence.